Hello, my name is Dr. Tobey Betthauser, and I am a RAP researcher at the University of Wisconsin-Madison Alzheimer's Disease Research Center. My research focuses on understanding the sequence of events that occur in Alzheimer's disease using Alzheimer's disease biomarkers. This includes both imaging and fluid biomarkers. The overall goal of this work is to understand the usefulness of these biomarkers to be able to predict if and when people will experience deficits in cognitive abilities related to Alzheimer's disease. This is particularly important during the stage of disease where proteins are accumulating in the AD brain, but people may not yet be showing overt cognitive symptoms. Before we get into the research results, I want to show you what positron emission tomography, or PET images, look like in people with and without Alzheimer's disease. In Alzheimer's disease, there are two sticky proteins that build up in the brain, plaques formed from beta-amyloid protein and neurofibrillary tangles formed from tau protein. PET imaging gives us the ability to see where and how much of these proteins are built up in the brain once there is enough of these proteins present that the scanner can detect them. In this slide, the images on the left side of the screen show what these PET scans look like for someone without detectable levels of amyloid and tau proteins. The amyloid scan is on the top, and the tau scan is on the bottom. The images on the right side of the screen show what these scans look like in the brain of someone with Alzheimer's disease. Comparing the amyloid scans, we can see that the signal is higher throughout most of the brain in the AD case, which is indicated by the more red color. If we focus our attention on the tau scans, we can see that there are distinct areas of the brain where the tau signal is higher in the AD case compared to the normal case. Understanding where and when these proteins deposit in the brain is crucial for understanding their role in Alzheimer's disease and how well we can possibly predict if and when someone will develop cognitive symptoms due to Alzheimer's disease. It is important to note that in the clinic, these scans are rated as either positive or negative by a radiologist. There are currently 3 amyloid PET tracers approved by the FDA for limited clinical use, and more recently, the first ever tau tracer was approved for clinical use. Thanks to the many dedicated RAP participants enrolled in longitudinal imaging biomarker studies, such as PREDICT, we have developed advanced statistical methods that allow us to estimate when someone became amyloid positive. In this recently published study, we show how we can estimate how long we think people have had amyloid in their brain based on their amyloid PET signal. In the plots on this slide, each line represents an individual participant's amyloid PET signal over time, with the red cases indicating those that are amyloid positive at some point during the study. The plots on the left show these patterns when looked at with respect to a person's age. By applying the statistical model indicated by the red arrow in the center of the screen, we are able to see that people who are amyloid positive tend to accumulate amyloid at about the same speed over time. This key observation allows us to take a single amyloid PET scan and estimate when someone became amyloid positive. Then, by subtracting a person's age at a study visit from this estimated age of amyloid positive, we can estimate how long someone has had amyloid in their brain at any given procedure. This, importantly, allows us to place people on a disease timeline rather than an age timeline. By applying this concept to cognitive data acquired since the beginning of the RAP study, we are finding that how long a person has been amyloid positive matters for their cognitive performance. In this figure, we're comparing the cognitive performance over time in people that were not amyloid positive, which is the black group, people that became amyloid positive at age 69, which is the red line, people that became amyloid positive at age 62, which is the green line, and people that became amyloid positive at age 51, which is the blue line. We can see that until about age 65, these lines look mostly the same. But as we get to age 70 and beyond, we can see that people that become amyloid positive earlier in life have worse performance and decline faster than those people without amyloid in their brains. We can also apply this concept to look at the timing of events in Alzheimer's disease. In this slide, we can see that people who have been amyloid positive for about 10 years are more likely to have higher levels of tau in their brain, and that for longer durations of amyloid positivity, we see even more tau in the brain. So from this study, we know that people with higher levels of amyloid on their PET scans have had amyloid for longer, and that people that have had amyloid for longer decline faster in cognitive performance and are more likely to have higher levels of tau in their brain. But this study did not look at how both amyloid and tau contribute to cognitive performance over time. In this next recently published study, we looked at just that. We used people's amyloid and tau PET scans to determine if they had elevated levels of amyloid or tau protein in their brains. With people sorted into amyloid and tau positive and negative groups, we then looked to see if cognitive performance was different between these biomarker groups in the 8 years leading up to their PET scans. What we observed was that people with elevated levels of both tau and amyloid, the group shown in the top right plot, had declined about 3 times faster in their cognitive performance during this 8-year period. This study is an important addition to the Alzheimer's disease research field, as it is one of very few studies to show this relationship between cognitive decline and AD biomarkers in people that were not cognitively impaired at the beginning of the study. And unique to WRAP and PREDICT, this study includes people that have been followed since their mid-50s, which is likely an important time for understanding how these early changes in the brain will impact cognitive abilities later in life for people with Alzheimer's disease. In this final slide, we combine these two research results into a movie that is meant to show the average pattern of Alzheimer's disease. The plot on the left shows cognitive performance over time for a stereotypical amyloid negative case, shown in blue, and a stereotypical Alzheimer's disease case that becomes amyloid positive at age 63, shown in red. The red star on the plot indicates when the Alzheimer's disease case becomes amyloid positive. The images on the right side of the screen show the average patterns of amyloid and tau accumulation in the Alzheimer's case. The images on the left side show amyloid accumulation looking at the brain from 2 different angles, and the images on the right side show tau accumulation from those same 2 angles. As the movie begins, we see that the cognitive performance is similar initially between these 2 cases. However, as the Alzheimer's disease case becomes amyloid positive, we see a faster rate of cognitive decline in this case, and we also see amyloid continue to accumulate in the brain. Once the AD case reaches the level for clinical impairment, as depicted by the dashed line in the left plot, we start to see increases in tau accumulation. And as we move forward in the disease, we see that both amyloid and tau continue to accumulate in the brain. It is important to point out that these studies and this video represent the average pattern of Alzheimer's disease and do not reflect the outcomes in individual cases. As a research field, we still lack the ability to take this information from PET scans and predict if and when someone will develop tau or other pathologies, and if and when someone will become cognitively impaired. This is a very active research area in WRAP, and it requires the continued commitment of study participants in imaging biomarker studies. As WRAP investigators, we are so incredibly fortunate to have such a committed group of participants that have allowed these major research developments to take place. As investigators, we feel strongly that this commitment and the contributions of WRAP participants will continue to provide key insights into the development of Alzheimer's disease during a critical time in the disease process. It is our hope that these findings will lead to better diagnostic accuracy for Alzheimer's disease during life, and that this work will ultimately contribute to a better understanding of how and when to treat Alzheimer's disease. I hope you enjoyed this video and found it informative, and thank you all for everything you are doing to contribute to the WRAP study.